Hello, everyone, and welcome. So today we're going to be covering protists and fungi. So two of the more diverse groups of eukaryotes on this planet. So let's dive in. Let's first talk about the origins of eukaryotic cells. So as we've discussed previously, eukaryotic cells are much more complex than our prokaryotic cousins, right? Thinking about our bacteria as well as our archaea. Now, eukaryotic cells are very different, as you'll probably recall from biology 1. They have exoskeletons, as well as this idea of compartmentalization. So we're thinking about membrane-bound organelles here, which makes them, again, very much more complex than their bacterial cousins. Now, the appearance of eukaryotic organisms in the fossil record seems to date back to about 1.5 billion years ago. As we can see in this picture down below, what appears to be a eukaryotic cell, relatively large, again, significantly larger than our bacterial friends. Now, when we're thinking about how compartmentalization arose, i.e. organelles, we actually know a little bit about how the endoplasmic reticulum and the nucleus came to be. Now, what we think happened here is that if we had a prokaryotic cell, we have some sort of infolding of the plasma membrane, which likely led to the nucleus as well as the endoplasmic reticulum. Now, we know this by looking at the lipid composition of our eukaryotic organelles. They very much resemble, at least for the nucleus and the endoplasmic reticulum, very much resemble the cell membrane. So that gives a lot of evidence to the fact that that's how they arose. But for our other two big organs, where we have our mitochondria and chloroplasts, we believe these have evolved one time or multiple times across the eukaryotic tree of life via this process of endosymbiosis. So the idea with endosymbiotic theory, actually, interestingly enough, for those of you that are from Massachusetts, you'll recognize that UMass Amherst, which is a sister school to UMass Boston, which is here in the Boston area. There's a scientist that works there. Her name is Lynn Margulis. She actually developed this theory of endosymbiosis. So a fun regional fact for those of you that are from Massachusetts. Now, endosymbiosis basically tells us that mitochondria and chloroplasts were basically arose via the symbiosis between a small, larger bacteria. What the idea being here is that we have this ancestral eukaryotic cell. It has that internal membrane system of the nucleus as well as the endoplasmic reticulum. An aerobic bacteria was engulfed by this, and over time, through this idea of endosymbiosis, symbiosis meaning a relationship, endo meaning inside, eventually caused this bacteria to evolve reductively to become a permanent association, thus leading to our beloved mitochondria. Then for our photosynthetic organisms, i.e. those with chloroplasts, those would have evolved through a symbiosis with a photosynthetic bacteria. Thus, over time, with reductive evolution, leading to an endosymbiotic event, giving us chloroplasts. So the idea here is that we have an endosymbiosis event. Now, it might be one event, it might be multiple events. Now, the presence of mitochondria across all eukaryotic organisms tells us there was only one event for the symbiosis of mitochondria. However, we know that due to different types of algae, such as red algae or brown algae, there likely was multiple symbiosis events, giving rise to sort of the distinct types of chlorophyll and the distinct photosynthetic structures we see across algae. Now, we know endosymbiosis is a thing, and it is pretty heavily supported by science at this moment in time. So we do know that the DNA inside chloroplasts and mitochondria are highly similar to those in the bacteria, both in size, both in shape, as well as the way the genes are transcribed. We know that the ribosomes found in mitochondria are very similar to bacterial ones, as well as chloroplasts and mitochondria replicate independently of mitosis, i.e. cellular division, by binary fission, very much the way that bacteria do. So we do know, again, very heavily in the case of endosymbiosis, that this is definitely how we give rise to mitochondria and chloroplasts. So pretty cool line of evolution. Again, some cool species interactions giving rise to complex organelles. Now, mitosis, as I mentioned, is something that eukaryotes do, and this is something that evolved inside eukaryotes. Remember, bacteria divide by binary fission. Now, prokaryotes, unlike eukaryotes, have one single chromosome. Again, eukaryotes having multiple chromosomes. And mitosis and cytokinesis had to develop to separate chromosomes and other cellular contents during cellular division. Again, a problem bacteria and archaea simply do not have. So with that out of the way, that sets the stage for the sort of the evolving of eukaryotic life. But let's talk about the most basic of all eukaryotic groups, which are protists. Now, these are the most diverse of all four eukaryotic kingdoms. In fact, we think that there are well over 1 million species of protists out there, depending on who you ask. Now, the funny thing about protists is their definition is rather, for lack of a better word, disappointing. And their definition, as you can see here from the second point, is that these groups of organisms that we call protists are united simply on the basis that they are not fungi, not plants or animals. And they are certainly not bacteria because they are eukaryotic. So if it doesn't fit into one of these three categories of fungi, plants, or animals, they are protists. So you can see that's kind of an unfortunate thing, right? They're lumped in because they're not something. But as we'll discuss, they vary considerably in all aspects of their life. They're unicellular, some are colonial, others are multicellular, most are microscopic, but many are large, visible with the naked eye. They have all sorts of different body symmetries, and they have all forms of nutritional needs. So again, they're united simply by the fact that they are not something, which is kind of a weird thing, but that's how we group the protists together. Now, protists themselves are present in all of the 6 eukaryotic supergroups. Now, protists themselves are not monophyletic, they are paraphyletic, and they're present in all 6 of these supergroups as we see here. So let's take a peek at this phylogeny. So we have the bacteria, the archaea, and then we have our lovely eukaryotes over here. And you'll notice that many of these 6 groups here include other things such as land plants, fungi, and animals. So protists are spread throughout all these large groups. But again, you might see in cases of excavata or rhizaria, they have their own unique supergroups that nothing else is found in. So again, very diverse, grouped together not because of anything other than the fact that they are not plants, fungi, or animals. So now, protists have a wide diversity of cellular structures. Now, protists have very varying cell surfaces. They do have plasma membranes. They typically don't have cell walls, but they can have cell walls. And they typically have what we call extracellular matrices. And we see this commonly manifest in weird ways, such as the diatom silica shells. Now, protists can also form cysts, which are very dormant and resistant outer coverings. And it's particularly useful when they are used for disease transmission. Now, protists, like bacteria, can have locomotion via flagella. So remember, those are those whip-like structures that propel bacteria through media or the environment. They can also propel themselves through cilia, which are shorter and more numerous short appendages that basically flap like wings in their environment, propelling them forward. Or very much like we'll see with amoebas, they can have what we call pseudopodia or false feet. And basically they move like a slug. They extend their front part out, pull their back part out, extend their front part out, pull their back part out. Again, very much the way a slug moves. So now our protists can have differing levels. They can be autotrophic as well as heterotrophic. And very much like bacteria, we also notice that there are mixotrophic, i.e. those that are both photosynthetic and autotrophic at the same exact time. So lots of interesting metabolisms inside of our protists. Now, reproduction in protists is divided into 2 key categories. They can be either asexual or sexual. Now, most protists reproduce asexually. Many protists have the capacity to reproduce sexually, but will preferentially reproduce asexually. And the sort of interesting thing is protists, some protists are strictly sexual in reproduction. So it's kind of this very varied thing. Whereas all mammalian organisms, for instance, reproduce sexually, protists again have this large amount of variability. Now, some species have some really weird mitosis. So some divide by mitosis, the way you sort of learned it in biology 1. Others divide by budding. Other divides by schizogony, which is cell division that is preceded by several nuclear division, producing several individuals. So again, a wide variety of asexual reproduction here, and sexual reproduction too, as we will discuss. Now, protists also form this bridge to multicellularity. Now, they are single cells typically, but they can form really interesting, complex colonies of microorganisms, and they can also form true multicellularity, as we'll see. Now, the interesting thing about multicellularity, it seems to have arisen multiple times. And as you guys probably know from your organ system, multicellularity fosters specialization, i.e. my liver does something very different than my brain, right? That's the level of speciality that you get with multicellularity. So, which is a pretty cool thing. So again, protists really provide that bridge between our bacteria and our archaea, and as well as the eukaryotic world. But again, very diverse, Very interesting group of organisms, and one you probably don't quite think about very much. Right? So let's dive into the first large group, which is going to be the excavata, which includes diplomonads, parabasalids, and euglenozoans. Sorry about that. And these share very much cytoskeletal features as well as DNA sequences. So let's talk about this group of organisms first. And we're going to walk through the rest of these groups as well. And eventually, at the end of the class, we'll talk about fungi. And then in the coming weeks, we'll talk about plants and animals as well. So kind of filling out this eukaryotic tree of life over the next few weeks. Now, diplomonads are unicellular, and they move with multiple flagella, as you can see here. Now, a great example of our diplomonads is actually Giardia. Now, Giardia is a waterborne parasitic infection you can get in the tropics by drinking stagnant water. You can also get it in non-tropical places too, but it's pretty heavily known to cause disease in the tropics. So it's kind of a nasty thing. Now, it's interesting about Giardia, as many diplomonads, they actually lack a functional mitochondria, which again is kind of a weird thing, right? They lack mitochondria, and they also have two nuclei. So they have this kind of weird metabolism where they don't rely on the electron transport chain, but again, they rely on other forms of metabolisms. Now, parabasalids are an interesting group of organisms. Now, the best known parabasalids are actually those that live in termite guts. So termites, as you may or may not know, eat wood. That's their primary nutrition source. And they've evolved this symbiotic relationship with both cellulose-degrading bacteria as well as cellulose-degrading parabasalids. So a really interesting group for the ecology of termites. Now, there's another one, Trichomonas vaginalis. As you can imagine, given its name, it causes an STD. So two really important groups. One's an STD. One is a symbiosis with one of the most important arthropods on the planet. Now, they have this really interesting undulating membrane for locomotion, as well as the capacity to use flagella, as you can see here. And their mitochondria is semi-functional. So they have a mitochondria. It's not fully functional, but again, it is semi-functional. So you can sort of see how we're going here, right? We're starting excavata. We started here with no mitochondria. Now we're at parabasalids, where we have a slightly functioning mitochondria. And so we're basically going to start building in complexity over time. Now, the euglenozoa, this is the one you might have heard of the most in terms of a protist. You might have heard of euglena. They're from euglenozoa. Now, they're an interesting group of organisms. Their bodies can actually change shape when they're swimming, alternating between being stretched out and rounded up. So it's kind of a cool thing. Now, they can also change shape because they lack cell walls. So they don't have that rigid outer structure that gives them that level of plasticity. Now, they're interestingly enough, they're among the mitochondria, and they're also free living as well as parasitic ones. Now, a third of all euglenids have chloroplasts and thus are autotrophic, while others lack chloroplasts, thus they ingest their food and are heterotrophic. Now, the reproduction is asexual, occurring via mitosis, and they, as I mentioned, they have flagella. They're actually anterior, what we call unequal flagella. Now, they have what we call contractile vacuoles, which allows them to collect excess water. And they also have what we call a stigma, which allows their movement towards light. So they have what's called phototaxis. So they're photosynthetic, and they can swim towards light because, as you can imagine, if you're photosynthetic, you need light to photosynthesize. And the other thing they have is, interestingly, they have many small chloroplasts, which we think have evolved from a symbiotic relationship through the ingestion of green algae. So we can look at our euglenids here. We have their structure here. You can see their really long flagella here, but you can see their overall structure. They're contractile vacuole. They have nucleus, chloroplasts, and things like that with their flagella. So this is our euglenids. This is what they look like. Now, within this group, we also have what we call parasitic kinetoplastids. Now, this is a really interesting group of organisms. They have a single mitochondria. And the weird thing is their DNA forms what we call maxi and mini circles, which are actually responsible for rapid glycolysis and really unusual RNA editing patterns. Now, the group we know from this are actually what we call trypanosomes. Now, for those of you that are interested in disease, you might recognize trypanosome because it causes 3 common diseases. First is African sleeping sickness, and it's carried by the tsetse fly. The second is leishmaniasis, which is a skin infection, which is caused by the sandfly. And then finally, Chagas disease, which is which is again a skin infection, which is caused by the contact with the blood or urine of an infected animal. Now, these are really difficult to control organisms, as most eukaryotic pathogens are. And the reality is that most things that are similar to us, i.e. eukaryotic pathogens, are hard to kill because they have very similar biology to us. Now, if you think how antibiotics work, they target bacterial biology. But if your biology is really similar to your pathogen, it would also target your own biology. So parasites like this, and as we'll see with fungi, are kind of hard to get rid of. However, our kinetoplastics are really interesting because they have this outer protective coat on them. It's not a cell wall, but it's an outer protective coat. Now, they have this really cool genetic mechanism that allows them to change the antigen, or essentially the surface proteins on their outer coat, thus making them more adaptable to the immune system and also much harder to control potentially with a vaccine. So a really cool and wild group of organisms. So that rounds out our tour of the excavata. There's a lot more there that we didn't cover, but I wanted to cover the most sort of biologically and medical relevant groups in that supergroup. So the next group is the chromalveolates. Now there are two distinct groups, the alveolata and the straminophila. And these are sort of interesting groups of organisms, as you'll see. They comprise many things you may or may not have heard of. So for instance, if you've heard of dinoflagellates, i.e. red tide, you've heard of chromalveolata. Sorry, it's a hard one to pronounce when you're talking for an hour straight, so I apologize for that. Now, it's interesting. They seem to have arisen by one or more secondary endosymbiotic events. So let's start with the alveolates. Now, these have flattened vesicles called alveolates. So we can see their sort of overall structure here. We have the alveolar sac with the apical complex. And again, you see their overall shape very, very much flattened. Now, the most common group we see from the alveolates are both the dinoflagellates, as you see here, as well as the acomplexians, as well as a group called the ciliates. Now, so that's our group here. Now, we'll dive into the next group, which is our straminophiles, which are brown algae, diatoms, and oomycetes. Now, it's a really interesting group of organisms. They have very fine hairs on their flagella, which is sort of an interesting thing. Not many organisms have very fine hair on the flagella, as we do see here with our straminophiles. Now, the other group is our dinoflagellates or dinoflagellates. Now, these include some pretty interesting groups of organisms. They're typically photosynthetic, living in aquatic and marine environments, and some are luminescent. So, for instance, if any of you have ever been to Puerto Rico, they have those biobays, those fluorescent bays that glow at night. Those are due to dinoflagellates as well. Now, the interesting thing about dinoflagellates and have histones. As you recall from Bio 1, our DNA and all the DNA of most eukaryotes is wrapped around histones. They actually seem to lack histones. So sort of interesting thing about them. Now, dinoflagellates as a whole simply seem to have chlorophyll A and C, as well as accessory pigments called carotenoids. And their biochemistry actually seems to resemble that of diatoms and brown algae as well. Now, they are primarily asexual, but they do undergo sexual reproduction under starvation conditions. And dinoflagellates are best known for actually an economic effect they have. So for those of you that have ever heard of red tide, so friends, have you ever gone down to the Cape and fishing or swimming is closed because of a red tide? Well, you can thank dinoflagellates. As their population explodes, and they get really high density, they produce toxins, and these toxins essentially build up in the water, killing, as I've noted here, fish, bird, and marine mammals, as well as you if you drank or red tide. So that's our dinoflagellates, a pretty cool group of organisms. Now, next up is our acomplexants, which are spore-forming animal parasites. So they're not the most unique of them, but they are certainly one of the most medically relevant groups on the planet. Now, they have this really weird structure where they have this pretty much all of their organelles at one end of the cell, which allows them to invade the host. So if you remember back from our first slide on this group of organisms, we were showing an acomplexan invading a host with all of its organelles on one side. Now, this is a really important organism, and you might ask, so why? And in fact, the genus of acomplexans called Plasmodium, which causes the disease malaria, is actually an acomplexan. So malaria is one of the most common infectious and deadly diseases on our planet currently, with about 85 million cases in Africa and tropical areas worldwide. So it is one of the most common and deadly diseases that we know of at this time period. Now, they have a really interesting life cycle. They have sexual, asexual, and different hosts. So we can actually look at what this looks like. So that we start off here with a malaria parasite living inside of a mosquito. Now, a mosquito feeds on a human and it injects the plasmodium, what we call sporocytes, and these go into the host liver. Now, once inside the host liver, these sporocytes invade and they go inside the liver cells and they hatch, essentially, not really, but sort of, producing what we call merozoites or merozytes. Now, these multiply inside red blood cells, and they kind of do this repeating over and over and over again until eventually, when certain merozytes develop into gametocytes, which then can infect a mosquito if a mosquito feeds on someone with malaria. Now, what that ends up happening is they differentiate into different types of gametes, fertilize, form an oocyte, then form the sporocyte, thus repeating the process over and over again. So again, this is what malaria does. Again, a really complex life cycle, right? We have two hosts involved, sometimes more hosts for certain plasmodiums. However, interestingly, again, a level of potential sexual reproduction, really, really unique group of organisms. Again, also a very deadly group of organisms. So now we have this next group, which is called the gregarians. Now these organisms, they're not particularly remarkable, but they do use their apical complex to attach themselves in the intestines of arthropods, annelids, as well as mollusks. So they are animal parasites. Now this leads us to another organism, which we call Toxoplasma gondii. Now, interesting group of organisms. This is a human pathogen as well. Now, it uses its apical complex to invade epithelial cells inside the human gut and causes infections of those that are immunosuppressed. And so it's actually a really common infection for those that have HIV. And it's kind of a hard thing to get rid of if you don't have a functioning immune system. And the other thing, Toxoplasma can actually cross the placental barrier to harm the fetus. So just as a note, if you've ever heard of like crazy cat ladies, one of the things that many crazy cat ladies, i.e. those that have dozens or sometimes hundreds of cats in their house, they're actually infected with this Toxoplasma parasite. And one of the things it does is it can actually cross into your brain, thus affecting your brain chemistry, thus affecting your behavior. So if you have a cat, be on the lookout for touching it to not touch its poo-poo or pee-pee, to not get Toxoplasma as a parasite. Now, this leads us to our next group, which we call the ciliates. Now, these are a really interesting group of organisms. They're also very beautiful. I recommend Googling microscopic images of ciliates. Again, a very beautiful group of organisms. Now, these have a large number of cilia arranged on longitudinal rows or spirals around their cell, and they beat in a coordinated fashion to propel the organism through its liquid environment. They also have a pellicle, which is a tough but very flexible outer covering. Now, they have two distinct types of nucleus, which makes them unique, a micro and a macro nucleus. Now, the micronucleus is only used for basically sexual reproduction, and the DNA is actually not transcribed. And then they have their macronucleus, which is your standard garden variety nucleus, i.e. transcription and mitosis and all that fun stuff. They also have two distinct vacuoles, a contractile vacuole, which allows them to regulate water balance, as well as a food vacuole, which allows them to digest food. And this is what we have, our ciliate here. You can see all the lovely little hairs. We have their food vacuole, we have the contractile vacuoles, two of them, the outer pellicle, and then they have their nucleuses here at the center. So really interesting group of organisms. And the really interesting thing about our ciliates is they can actually do a process called conjugation, i.e. direct cell-to-cell contact for the exchange of genetic information. Now, the interesting thing about these organisms, it's a really similar process to bacteria, right? It's direct cell-to-cell contact with the exchange of genetic information. However, for them, it's actually the exchange of their micronuclei. So you can go through this diagram and take a peek at it. But again, similar to the process of bacterial conjugation, just a different item being exchanged. So I think that's actually a really cool way to reproduce sexually as an organism. Now, our final group is going to be from our stromatophiles is going to be our brown algae, which are seaweeds. So I'm sure you guys have probably seen pictures of seaweeds at some point in time, or maybe you're at the New England Aquarium or another aquarium and you've seen brown algae somewhere. They're common across the northern hemisphere and they can get quite big. So for instance, off the California coast, they have those 30-meter tall kelp forests, which are brown algae. Now, they have a really important life cycle, and you're going to want to remember this life cycle, and you're going to want to remember this name, this haplodiplonic life cycle, which involves the alternation of generations. And I say you're going to want to remember this because you're going to want to know it for protists, you're going to want to know it for fungi, and you're going to want to know it for plants. So remember haplodiplonic, alternation of generations, alternating between a sporophyte, i.e. multicellular, as well as a diploid phase, and a gametophyte phase, which is multicellular and haploid. Now, the important thing to mention about our brown algae is they look like plants, but they are in fact not plants. So they look like it, but again, they're not. Now, what does a haplodiplonic life cycle look like? Well, this is it. We'll start off with sperm and egg. Sperm meets egg and a fertilization event occurs. Now, as you recall from Bio 1, sperm is haploid, egg is also haploid. When you fertilize, you get a zygote, which is 2N. Now, their zygote develops into a developing sporophyte, which then in turn develops into a fully mature sporophyte, which is basically the fully grown brown algae that we see. So its full adult form is a sporophyte 2N. Then meiosis occurs, producing zoospores, which then germinate and differentiate into gametophytes, which are haploid. And now those different gametophytes are either egg or sperm, and then the process repeats itself over and over again. So again, you definitely want to remember this idea of haplodiplonic life cycle, this dichotomy between haploid and diploid organisms. Now, next up, we have our diatoms. Now, our diatoms are a pretty cool group of organisms. Now, they are in the phylum Chrysophyta. They are photosynthetic, they are unicellular, and they are also one of the most remarkably crazy-looking protists under the microscope. So these are some pictures down here at the bottom, but I do recommend taking a peek because, again, it is a really wild group of organisms under the microscope. It's a great thing to sort of look look on Google, something like that, to find some more pictures, because again, they're really a beautiful, beautiful group of organisms. So now they have really unique double shells that are actually made of silica, and some actually use what we call raphines, which are two long grooves lined with vibrating fibrils, which we see here. So these are diatoms. Again, really cool photosynthetic organisms. Just from these pictures here, right? They're really, really beautiful. Which leads us to our next group of, which we call the oomycetes, which are what we call water molds. Now, they are typically what we call parasites or saprobes. Saprobes are just things that like to eat dead material. So that is our oomycetes. Now, they were actually originally considered fungi, but they're actually been revised to be inside the Protists lineages as well. Now, they reproduce asexually, and they have what we call motile zoospores that have two unequal flagella. And so they do undergo some level of sexual reproduction, and they are found in soils. They're also found on lands. Now, there are many types of oomycetes that have been known to cause pathogens, so including Phytophthora, infestans, which actually caused the Irish potato famine, which killed about 500,000 people. And we also see modern oomycete pathogens that harm our crops, such as soybean and corn and things like that. So again, major groups of agricultural pathogens, including ones that have killed hundreds of thousands. So that's our chrome alveola. This leads us next to our next group, which is we call the Rhizaria, which use pseudopods or false feet for locomotion. And when there are three distinct ones we're going to talk about here: the Radiolaria, the Foraminifera, and the Cercozoa. Now, the Radiolaria are, again, a lot of really beautiful protists out there, but this is probably my favorite. The Radiolarians have this glassy exoskeleton made of silica and needle-like pseudopods or false feet. And you can just see how this just really looks like the sun, right? I mean, this organism, it's green, but it looks like our sun. If you Google a picture of the sun, this is what the sun looks like, but again, different colors here. So again, really, really beautiful. Now, this group of organisms, the Radiolarians, they're heterotrophic, in particular the Foraminifera, I should say, are heterotrophic marine protists. Now, they have these pore-studded shells called tests, through which thin podia emerge, and the podia themselves are used for swimming and feeding. So we can see the pseudopodia here poking through those little test holes there. And as I mentioned, these are made out of silica. So for those of you that have ever heard of the Cliffs of Dover in England, the Cliffs of Dover in England are actually just dead and calcified silica shells of all these Foraminifera just piled on top of one another. Now, for those of you that have never heard of the Cliffs of Dover, this is just a small picture of them, but they are these beautiful white cliffs off the UK coast. I forget exactly where off the top of my head, but again, it's from the UK. And again, all these white cliffs are just dead silica Foraminifera piled on top of one another, again, creating this beautiful, beautiful landscape that we see off the UK coast. Now, our next group is our Cercozoa, which is a very morphologically group of soil protists. Now, they use locomotion via flagella or pseudopods, and many also have silica-based shells made of scales or plates. So that's our Rhizaria. Not a lot of interesting stuff in that group, not a lot of wacky and wild stuff outside of the cliffs of Dover, but certainly a pretty group of organisms. Now, the next group is going to be the Archaeoplastida. Now, this group consists of Rhytophyta, chlorophytes, caryophytes, as well as land plants. And land plants are going to be the discussion of our next week's lecture. Now, these are photosynthetic organisms, again, acquiring their chlorophyll and their chloroplast via endosymbiotic events. Now, Rhodophyta, it consists of our red algae. Now, red algae are really kind of a funky group. Many of them are microscopic and single cellular, but others are very, very large. Like you can go find red algae off the coast here in Massachusetts. Now, interesting of our Rhodophyta, they lack flagella, but they also lack centrioles. So you guys might recall centrioles are involved with the production of microtubules. So, and our Rhodophyta also have many accessory photosynthetic pigments, which give them their red color, i.e. why they're red algae. Now, this is what red algae looks like. Again, they range from microscopic to very, very large, as we see here. And they have a large variety of cell shapes, shell structures, and overall 3D shapes to them. But again, they're unified in their red color and their photosynthetic pigments that they have. Algae, in my opinion, but they're comprised of two main lineages: the Chlorophyta and the Caryophytes. So let's talk about those. But I just want to make mention before we move on is that the Caryophytes actually gave rise to the land plants. So we'll talk about that next week, but definitely an important group for plants. Now, the unicellular chlorophytes, they're the very early green algae, and they actually likely resembled an organism which we call Chlamydomonas reinhardti. It's actually a model organism for understanding algal biology. And they did diverge from land plants about 1 billion years ago or so. And there seems to be many evolutionary specializations that have evolved from chlorophytes as well. But again, they have this haplodiplonic life cycle. We have a fertilization event occurs, producing what we call a zygospore, leading to meiosis, producing two types of gametes. Now, instead of male and female, it's actually plus and minus. And then when the plus and minus make it together after they developed and undergo asexual reproduction, they fuse minus and plus, pairing them together, allowing fertilization to occur, repeating this process over and over again. So again, a haplodiplonic life cycle here. Now, there's also some really interesting cell specializations in our colonial chlorophytes. Now, as I mentioned, multicellularity seems to have arisen multiple times across eukaryotic organisms. However, there are colonial chlorophytes out there. So, for instance, Volvox, which has a hollow sphere, as you can see here, and this hollow sphere is actually comprised of 500 to 60,000 individual cells, each with 2 flagella. So it's kind of a wild organism, Volvox. Again, also a very pretty organism. Now, as I've mentioned, the haplodiplonic life cycle is also common in multicellular chlorophytes. And so we're going to talk about one which we call ulva. Now, this is a multicellular chlorophyte. It looks like green algae, right? It looks like seaweed almost, right? But it's not quite seaweed, but it does look like seaweed. Now, we can actually look at what this looks like. So we'll start off with fertilization here, which leads us to a zygote. So it's 2N, leads to a germinating zygote, which then leads to a sporophyte, which is 2N. Now, that's the full adult plant. But you'll notice in this diagram, there are 2 types of full adult plants. And we'll get to that in 1 second. Now, these 2Ns produce sporangia, which then develop via meiosis. So they cut their number in half, becoming N, developing into spores, becoming a plus gametophyte or a minus gametophyte. Then those, as you can see, are fully grown organisms, which then undergo a form of meiosis, leading to minus and plus gametangia, leading to minus and plus gametes, then leading to fertilization, and thus starting the cycle over and over again. So again, really distinct life cycle here. 2N has an adult form as well as N having an adult form. Now, our other group is the caryophytes. These are green algae, and they are distinguished by having a close phylogenetic relationship to land plants relative to the chlorophytes, which we just discussed. And we have 2 examples here, Chara and Cleidocete. And you can see they resemble pretty closely land plants. Thus, they are one of the relatives to land plants. Now, caryophytes have they have not the haplodiplonic life cycle, but a haplonic life cycle. Now, there are 2 candidate chlorophyte clades thought to be most closely related to land plants. These include the corales and the Calicocitales, sorry about that. It's a tough word to say when you're sort of in the middle of a lecture. But both of them seem to actually form green mats around the edges of freshwater ponds and marshes. And it seems to be that because of their sort of the way they grow, they might have eventually inched their way onto land, thus eventually becoming land plants. Now, another group within this, as we mentioned, is the amoebozoa. And you guys probably have heard of, oh, this person's as stupid as an amoeba, or they're simple as an amoeba, right? Used to be a pretty common insult back in the day. That, of course, assumes that you're old enough to know that that's an actual insult. Or maybe it was just a scientific insult. I don't know. I remember someone saying that once, so it always kind of stuck in my head. But regardless, amoebas, they move by means of pseudopods, or those false feet. And remember, if pseudopods, they extended out and dragged their butt with them. Extended out, dragged their butt with them. That's what pseudopodia does. But again, all a pseudopod is a false foot, but it's really just a flowing projection of the cytoplasm. And again, extending and pulling the forward, but it's also used to actually extend, engulf prey, and eat. And now this way they're eating, this engulfing is actually a similar process to the way your own white blood cells extend, engulf, and eat bacterial pathogens as well. So that's our amoeba, and they're their own sort of distinct lineage here, distinct from everything else. So they are, again, unique. Now, one group I'm going to talk about is actually amoeba Proteus. Now, these are pretty interesting looking organisms, but amoebas are free living. They're found in soil, and they're pretty common in fresh water. Many are parasitic. And so, for instance, acanthamoeba actually enters the body through wounds, crossing the blood-brain barrier and causing brain infections. So they actually eat your brain, which is kind of a frightening look into biology, but it's a pretty important one. Now, this leads us to the end of amoeba to actually talk about the plasmodial slime molds, which are really sort of this really wild group of organisms. If you want to know more about slime molds, we're only going to briefly talk about them, but there's a really cool one as part of the Paris Zoo. It's called Physarium. I recommend you Google it. It's Physarium, Physarium. Really, really cool slime mold. But let's dive into these slime molds. Now, they stream along the ground as plasmodium, which are non-walled, multinucleated masses of cytoplasm. And this is what they do to feed. So you can see this one is spread all over a log, and it's doing this to spread out and feed upon the log as well as the things on the log. They eat bacteria as well as organic food, and they actually form these sporangia, which kind of look like blueberries. At least to me, they look like blueberries, where they actually form spores and they reproduce by releasing these spores into the air. So that's our plasmodial slime spores. Now we have cellular slime molds, which actually really interesting group of organisms that we use to study cellular differentiation, because they have very, very simple developmental systems. Now these individual organisms actually behave as separate amoebas, but they move through the soil ingesting bacteria as individuals, and when food is scarce, they actually aggregate to form a slug, and these slugs themselves can make what we call spore cells. And you can sort of see this life cycle here. This is Dictyostelium dickosidium, which is a cellular slime mold. So it starts off as an individual slime mold. They come together to form this slug-like thing, which then allows them to develop further to a longer slug-like thing, leading to this complex where they produce these spores, reproducing, and then repeating the life cycle over and over again. But again, these are multicellular aggregates of single cells coming together and thus fusing, having this really interesting biological trajectory of themselves. Now we have the canoflagellates, which are the opisthokonts. I can never remember if that's how you pronounce it, but that's how I'm going to pronounce it. Now these are unicellular, and they're the actually closest relative to fungi and animals, and they're actually most likely the common ancestor of sponges as well. And we'll talk about sponges later in the semester. Now, these organisms have a single flagella surrounded by a funnel-shaped contractile collar, and this structure is actually matched pretty much structure for structure in our sponges as well, which again we'll talk about in a few weeks. So this is our I'm not going to try to say it. It includes fungi, us, as well as the canoflagellates. Now, colonial canoflagellates actually resemble their animal relatives, the sponges. Now, we'll see sponges in a few weeks, but sponges actually really look heavily like this. Circular, not a lot of the descript stuff, but again, very distinct organisms. Now, this leads us into our final group for the day, and it's not a protist, so we've just wrapped up our protists. But it leads us to actually probably the most exciting group of eukaryotes, at least in my opinion, which is actually the fungi. And one of the reasons they're really exciting is because they have this really interesting set of biology that makes them useful for nature as well as for us. So for those of you that like bread or beer or wine or kombucha, you owe a lot to the fungi. And again, they're really, really important. And we'll talk about all the amazing things fungi do. So mycologists, or those that study fungi, believe that there's probably upwards of 1.5 million fungi out there. Some people estimate more, but again, I think the baseline is probably about 1.5 million. So a lot of fungi. And to put that in perspective, there's about 1.2 million described species in total right now, and we project that there's way more fungi than that. Now, fungi can be single cellular or multicellular, sexual or asexual, and have some really weird mitosis. And they're also specialized to extract and absorb nutrients from their surroundings. And we split off from fungi about 460 million years ago. Now, it's not settled science right now, but it does seem to be around 460 million years ago. Now, fungal biology is hard. Fungal phylogenetics is equally as hard. But there seems to be 6 major monophyletic phyla, and these are listed here, and we're going to talk about all of these. And I'm not going to try to say the second one because it's a mouthful to say, but I'm just going to call it Neo when I get to it. And they also have one paraphyletic phylum, which is the Zygomycoda. And in addition, we also have some fungi, which are called the Microsporidia, are also included in this group, but not always. And they're fungi, but they don't seem to be fungi. And we'll talk about them later in the talk. So let's look at this. We have our tree of fungi here. There's some ancestor. We have the 2 most important groups of fungi over here, which we'll spend the most time talking about. And there's other groups here. Again, illy defined phylogeny of poorly understood organisms. But let's dive in. But I should make mention before we dive in, we have this handy dandy table. We have all the major groups of fungi, an example of them, their key characteristics, as well as the living number of species. But you'll notice that, as I mentioned, the most important ones, the Ascomycota and the Basidiomycota, have upwards of 30,000 species in them. So very, very diverse, whereas something like the Neo group here has only about 20 species. So certainly more fungi are more prolific and larger distributed and more diverse than others as well. Now, fungi have general biology that we can understand. It's not particularly very complex, but multicellular fungi consist of long, slender filaments, which we call hyphae. Now, hyphae can be continuous. Others can be divided by what we call septa, and I'll show you a picture of that in a moment. And their cytoplasma actually basically flows throughout their hyphae. You can think of hyphae as like roots, and you can think of their cytoplasm basically flowing through their hyphae, basically like a river. But we can look at what we have here. Now, this is a microscope picture here, but we have a hypha here, or hyphae, and we have the structure here. Now, we can see the septa here dividing this, but when there's no septa, it would basically just be like a hollow tube. Excuse me, hollow tube. But again, this is what our hyphae look like. They look very much like roots. And I think that's kind of a cool thing. You'll actually notice the septum here is actually analogous to a structure we'll talk about plants in our next lecture together. Now, fungi have what we call mycelium, which is a mass of connected hyphae, and it grows through it grows through and digests its substrate. So think about like a hyphae just penetrating a sandwich and then digesting the sandwich. That's basically what a mycelium does. Now, the fungal cell walls include a really important compound called chitin. Now, chitin, as we'll discuss, is actually found in the shells or the exoskeletons of arthropods, one of the most successful groups of organisms on this planet. But again, chitin is very, very tough. It's a macromolecule. It's a really abundant macromolecule because of how many fungi are on this planet, as well as how many arthropods. But chitin is really rigid, really tough, makes fungi a tough class of organisms. But we can see the individual hyphae here, and we can see how the hyphae grow on this bark, again, spreading out overall into this over this wood. So again, they grow throughout their substrate, spreading throughout, allowing them to grow. Now, hyphae themselves can have one or more nuclei. So they can be monokaryotic, which is one nuclei, as well as dikaryotic, which has 2 nuclei. And the interesting thing is that both genomes, if they're dikaryotic, are actually transcribed. So it's kind of a weird thing. Now, sometimes many nuclei actually intermingle in the common cytoplasm of a fungal mycelium, forming either a heterokaryotic or eukaryotic organism, which is where we have nuclei from genetically distinct individuals, or homokaryotic, which are nuclei that are genetically similar to one another. So you think about it like if I had a cell that had two nuclei, if both of them were my nuclei, I'd be homokaryotic. But if one nuclei was from my mom and the other one was my own, I would then be heterokaryotic. It's kind of a weird thing. Now, fungi also have weird mitosis. Now, their cell, for them, is actually not a really relevant unit of reproduction, but what ends up happening is their nuclei envelope actually doesn't break down and reform like we typically think for mitosis, but instead their spindle apparatus actually forms within their nucleus. So it's kind of a weird thing. Now, with the exception of the chytrids, which we'll talk about later on, fungi do lack centrioles. But again, they don't have the same mitosis the way we think. So let's dive into the reproduction. Now, they are capable of sexual and asexual reproduction. Now, their sexual reproduction involves the fusion of two haploid hyphae of comparable mating types. Now, for instance, in some fungi, fusion immediately results in a diploid or two end cells, while others have what we call a dikaryon stage, one end plus one end, before a parental nuclei forms a diploid nucleus. And many of our fungi, when they're sexually reproducing, actually form mushrooms or what we call puffballs. Now, inside mushrooms and puffballs, we have what are called spores, which are the most common means of reproduction among fungi. And you'll actually see this is the common way plants reproduce as well. Now, many asexual processes and are commonly dispersed via wind. Now, fungi have a wide variety of nutritional avenues to take advantage of, but the most common way fungi eat is actually by secreting digestive enzyme into the surroundings and then essentially absorbing the organic molecules via this external digestion. So the idea would be here that they vomit and then they suck up their vomit after it's digest their food, which again, it's kind of a gross thing to think about, but that's basically what they're doing. Now, the cool thing about fungi is they can actually break down relatively easily both cellulose and lignin, which, as you will probably recognize, are two components of the plant, of plants, in particular, two components of wood. So they are really good at decomposing wood and other plant parts. We also know there are many carnivorous fungi out there, as well as some parasitic fungi as well. So really cool organisms. Now, in terms of their ecology, fungi, like bacteria, are the principal decomposers in the biosphere. So they comprise what we call the carbon cycle, as we'll discuss later in the semester. Now, many are very, very difficult to isolate in culture. So it's really hard to understand a lot of fungal biology, but we know they're out there and we know they're instrumental for the function of biospheres as a whole. And they do, again, as they mentioned, they do break down cellulose and lignin, releasing carbon, nitrogen, and phosphorus into the world around them. So really cool role in nature. They also form some really interesting interactions with other species, including many symbioses. So they can form facultative or obligate symbiosis with other organisms on the planet. They can also interact with others, either as parasites or pathogens. They can also form commensal or mutualistic relationships as well. So they basically can fill any role in terms of a species-species interaction. Now, one I'd like to discuss, and it's actually one of the most important symbioses on the planet, is actually what we call endophytic fungi. Now, endophytic fungi live in the intercellular spaces inside the roots, as well as the shoots, but primarily we think about the roots of plants. Now, some of these fungi are parasitic, but most, and by and large, most endophytic fungi are commensal, i.e. they help their host. I'm actually going to skip this slide, but it's kind of interesting little experiment that you can definitely read on your own if you would like to, of course. Now, one of the more interesting symbioses out there is actually the lichen. Now, you've seen lichen out there. You've just never probably really noticed it. And I say you've probably never noticed it because you've definitely seen this if you've walked around the city of Boston, these foliose lichen or potentially these crustos lichen. Again, they are everywhere. They colonize bare spaces. And what's really cool about these lichen is their symbiotic relationships between a fungi and some sort of photosynthetic partner. Now, it's typically a cyanobacteria, but it can also commonly be green algae, and sometimes it's both. And these lichen can also have symbioses with bacteria. And these interactions are mutualistic, i.e. both organisms benefit. And typically speaking, lichens are what we call the ascomycetes. And and it's really, really common. So there's about 15,000 species of lichen. Ascomycetes are all but 20 of those 15,000 are ascomycetes. So pretty cool organisms. Now, fungi in lichens are actually, interestingly enough, are unable to grow without their photosynthetic counterparts. However, what the fungi do provide to their photosynthetic counterparts in this lichen symbiosis is protection, as well as preventing from drying out. So basically, they give a little house to their photosynthetic partners. And as you probably noticed, seeing lichen around in the Boston area, they live in some pretty harsh environments. So they grow anywhere. They have these really cool symbioses. They protect their partners, giving them a space, giving them protection, and their partners actually give them photosynthetic byproducts, i.e., food, in exchange for this housing. And the other thing about lichen is they're actually really sensitive to pollutants. Now, there are three groups of lichen: the fructose, the foliose, and the crustose lichen. We don't see these very much here in the greater Boston area, but we do commonly see foliose and crustose lichen spread throughout. And one of the reasons we don't see much of these fructose lichen is that they're very, very pollution sensitive. Now, the other type of endophytic symbiosis for lichen that's really important is actually the mycorrhizal mutualistic relationships. And these again occur between a fungi and a plant. Now, the really amazing thing about this is these mycorrhizal fungi are found on about 90% of all known vascular plant species on their roots. And basically what they do is their hyphae, which resemble roots, actually act as essentially as an extra rooting system, allowing for the uptake of extra nutrients from the environment, allowing plants to be essentially much more efficient at what they do. So again, 90% of all known vascular plant species, including all of our agricultural plants, have some sort of mycorrhizal association. So this association between plants and fungi is arguably one of the most important symbioses on the planet. So pretty cool. Now, arbuscular mycorrhizae are one type of these. Now, this is the most common that we know of in terms of these mycorrhizal symbioses. Now, the fungal partners in this symbiosis are glomeromycetes, where they actually don't produce any above ground fruiting structures. And they are, again, really important for agricultural systems, in particular in places where they have very low phosphate as well as low energy inputs. But what ends up happening is these arbuscular mycorrhizal fungi, their hyphae penetrates the root cell wall, but actually not the plant membrane. So this is a plant root here. This is a cross section of it. And you'll notice that the mycorrhizal fungi here, so our arbuscular ones, they penetrate the plant's outer root wall, and they essentially embed themselves inside the plant's, essentially, as we'll see soon, inside the plant's root, inside some of the cells. Again, you can see a nice microscopic picture of this as well. But again, these physically penetrate the plant roots, anchoring themselves, then extend away from the plant roots, providing an extension of the plant roots, allowing the plants to essentially get access to more nutrients, more water, and things like that. Now, this leads us to our next group, which is what we call the ectomycorrhizal, which are actually pretty common in forest trees, such as oaks and pines. And their partners in this ectomycorrhizae are plants, as well as a group of fungi called the basidiomycetes. Now, there's about 5,000 species of fungi that seem to be involved in ectomycorrhizal relationship. And their hyphae actually surround, but actually do not penetrate the root cells. So we'll look at this again. We have the outer root of the plant. This is just going to be a cross section. Lots of ectomycorrhizal fungi on the outside, penetrating the outer layer, but not directly invading the cells of the plant root. So similar to our arbuscular mycorrhizal fungi, but again, no penetration of the cells, but again, covering the roots, providing an extra layer of surface area for the absorption of nutrients, water, things like that. Now, this leads us to a really cool animal mutual symbiosis. So in many ruminant animals like cows, they actually host these this fungi, I'm not going to try to say it, in their gut, which aids them in breaking down of organic material. In addition, leafcutter ants have actually domesticated fungi, which they actually keep underground in gardens. If you've never heard of leafcutter ants, I definitely recommend Googling these and watching a few YouTube videos on them. They're wacky and wild. They're actually performing agriculture, right? What they do is they harvest leaves, They get fungi to grow on those leaves, and they tend their fungal gardens and feed on the fungi in this really, again, wild symbiosis. Now, so those are the helpful things fungi do. But as we've mentioned, fungi are parasites and pathogens. Now, they cause many, many health issues in humans, including allergens. So many fungal spores can cause allergic reactions, but as well as respiratory illnesses if they're ingested in high enough numbers. They can also form mucoses, which are direct infections, either on skin or nails. And also their toxins that they produce can actually be really nasty as well. So fungi have the capacity to be particularly harmful to us, but they're, generally speaking, not a super big deal. Now, fungi have been known to produce some wild secretions, which we call mycotoxins, which can potentially make food unpalatable, potentially carcinogenic, or even poisonous. So, for instance, Aspergillus flavus produces what we call the alpha toxin. And this fungi here actually produces a toxin which causes corn smut, which actually harms the plants, but is actually not toxic to us or the animals that potentially might eat these plants. Now, there are many diseases that humans suffer as a result of fungi. So for those of you that have ever had athlete's foot, that's caused by fungi. If you've ever had ringworm, it's caused by fungi. If you ever had fungus on your nail, it's caused by fungi. If you ever had if you're a woman, you've ever had a yeast infection in your downstairs area, caused by a fungi. So they are common pathogens, not as common as bacteria or viruses, but they still are common. And very much like protists, they're actually really hard to treat. Very much like protists, they have very similar biology to us, which makes our antibiotics, or I should say our antifungals and other medications really hard to develop because we don't want a medication to kill us in addition to killing our pathogen. Now, in addition, there's a really important fungal pathogen. It's caused by the organism Batrachotrypium dendrobatidis, which causes the disease chytridiomycosis, or chytrid for short. Now, this is from the phylum Chytridiomycota, and it actually causes an infection of amphibian skin. It causes this really nasty osmotic disruption in the skin of amphibians, which has actually led to a worldwide decline in amphibian populations. Again, all caused by this one singular pathogen. Now, this pathogen we'll actually talk about again later in the semester when we talk about conservation, but do know this pathogen, which is really bad for frogs and salamanders and other amphibians, is primarily trafficked around the globe via human activity. So a bad organism, but made worse by humans. And again, we'll talk about that later on. Now, many fungal diseases also cause infections on our plants. So there are many things that cause damage to living plants that we might eat, or harvested plants that are stored for later consumption as well. And they can also, actually, I'm going to skip this. We don't need to talk about that. So we're going to talk about our first group of fungi, which we call the Basidiomycota. Now, these are some of the most familiar fungi that you've probably seen. You've seen a lot of fungi, you just don't realize that you've seen a lot of fungi. Now, Basidiomycota include many mushrooms, as well as toadstools, puffballs, shell fungi, etc. For those of you that like Super Mario is based off of toadstools, which is a type of Basidiomycota. And I say toadstool as in the character Toad from Super Mario. Now, they also have some pretty important pathogens like rusts and smuts, and they're actually named for what we call their basidium, which is actually these club-shaped sexual reproductive structures. Now, Basidiomycota have a really interesting life cycle that we'll dive into. Now, they have this process called karyogamy that occurs within their basidia, which, again, how they get their name. And so let's look at this. So we have so we'll start off here at 2N, where we have a fertilized zygote, i.e. 2N, that undergoes meiosis, producing basidium, which have some interesting structures to them. But again, they kind of look, they look like little fingers coming off, at least in my opinion. Now, these basidium produce basidiospores, which produce either a minus mating strain or a plus mating strain, which allows them to essentially fuse together, having two distinct nuclei. But again, it's not like 2N, it's N plus N, so two distinct non-fused nuclei, which again, it's a result of the plus and minus mating strains fusing. That eventually forms a basidiocarp, or just a mushroom. Now, the gills underneath the mushroom are lined with basidia, which produce basidium, which then the two nuclei fuse in a process called karyogamy, forming that zygote, thus causing the process to repeat again. So again, this weird, again, it's haplodiplonic, right? 2N, but they have this weird N plus N cycle where they have two distinct nuclei in their cell, and they're essentially becoming 2N is just a fusion of those two nuclei in the zygote, of course. Now, spore germination in our Basidiomycodia does lead to production of monokaryotic hyphae, which results in monokaryotic mycelium or primary mycelium. Now, different mating types of monokaryotic hyphae may fuse, resulting in those dikaryotic mycelium or secondary mycelium, as we saw in the previous slide. And their basidiocarps, i.e. mushrooms, are formed entirely of secondary mycelium. So if you've ever eaten a mushroom, which if you don't eat mushrooms, I recommend you do. They're very, very healthy for you, but only eat the ones that are safe. Definitely don't go hunting mushrooms if you don't know what you're doing. But they are very nutritious, very, very healthy for you. Now, this leads us to our Ascomycota, which contain about 75% of all known functions. Now, this includes really awesome, economically important groups, including yeasts. So if you like beer, if you like bread, like I do, like I'm a bread fanatic, I love Ascomycota because they give me yeast, which gives me bread. But also other things, if you guys like blue cheese, they're Ascomycota as well. Cup fungi, truffles, and morels are all from the Ascomycota. Now, some of the most serious plant pathogens out there, including those that cause blight as well as Dutch elm disease, are from the Ascomycota. In addition, penicillin-producing fungi, so penicillin is just an antibiotic, one of the first mass-marketed antibiotics, comes from Ascomycota in the genus Penicillium. Fun enough, Penicillium as a genus doesn't just produce antibiotics, it also helps produce tasty cheese. So if you like cheeses like Camembert, you like Ascomycota. And now these groups are named for their ascus, which are microscopic sac-like reproductive structures. So let's dive into this organism. So we'll start off with meiosis. So we have 2N, which leads to an N organism. Then mitosis takes over, eventually producing a structure that produces ascospores. Now these ascospores then derive a plus and a minus organism, and these actually can reproduce as mycelium by producing candida, which allows them to essentially continuously make their mycelia longer, as we see in the minus as well as in the plus strains as well. But when their plus and minus strain fuse via plasmogamy, this produces an N plus N organism, producing what we call dikaryotic hyphae, which then develop into an ascocarp, which is similar to a mushroom, but again, very, very distinct. Looks more like a cup or more like one of these kind of funky brain-like looking structures, which then leads to ascus being formed, which then undergo karyogamy, remember the fusion of nuclei, thus leading to a 2N organism, thus repeating the process over and over again. So wild and wacky life cycle. Now, within the Ascomycota is our yeasts, which are single-celled, and they typically reproduce sexually by cellular fission or budding. And we like these because they ferment carbohydrates in ways that are delicious, including breaking down glucose to make beer, bread, and wine. And if you've ever heard of Saccharomyces cerevisiae, it is an Ascomycota yeast. Now, yeasts are also used as a pretty awesome model organism. We know so much about human biology from studying the biology of these single-cellular yeasts, in particular, our beautiful friend here, Saccharomyces cerevisiae. Now, this leads us to our next group of fungi, which are the glomeromycetes, which are very, very small. Now, these ones, as we mentioned, form symbiotic associations with the plant roots called arbuscular mycorrhizae, and really interesting group of organisms because they actually can't survive in the absence of a host plant. And they don't seem to reproduce sexually, they seem to only reproduce asexually. Now, this leads us to our next group, which is the zygomycetes or the Zygomycota, and they're incredibly, incredibly diverse. Now, unlike the rest of our organisms, they are not monophyletic. They seem to be paraphyletic, but we're still not quite sure. It's kind of an area of active research for us. Now, these include some pretty common bread molds. So if you ever had mold on your bread, you can think of Zygomycote for that. And we also know a few human pathogens as well. Now, let's dive into their sexual reproduction. So we have, we'll start at 2N like we always do. We have meiosis that produces N, but they produce these germinating zygosporangium, which then undergo germination to produce spores. Now, these spores are released onto their media. In this case, it's bread, which undergo asexual reproduction to propagate on the environment. And eventually, they produce rhizoids, which allow them to spread out easier. But they have a plus mating strain and a minus mating strain, as we've seen thus far. So you can think of those almost like male and females, which then produce plus and minus hyphae, which then form what we call the gametangia, which fuse together under a process called plasmogamy, as we've seen, producing an N plus N organism. Then karyogamy undertakes, fusing the two nuclei together, forming 2N, thus allowing them to come together. So it is, in a way, it's kind of a funky way of sexual reproduction, but again, it's just an organism producing offspring with itself. Now, this leads us to the chytridiomycota. Now, these are typically just commonly called chytrids. Now, these are commonly found in aquatic and marine environments, and they're typically flagellated, and they're very, very closely related to the most, what we think to be what the original fungi looked like. So these are a very ancient group of organisms. And we've talked a little bit about, we talked about this organism here, Batrachithium dendrobatus, as a mass murderer of all amphibians. We talked about that a few slides ago, so we won't dive too much more into the chytrids as well. But I do want to show you a picture. So this is an adult chytrid. It produces these zoospores, which then go swim out and undergo a level of sexual reproduction, as we've seen before. Now, this leads us to our next group of organisms, which we call the blastocleidomycetes, which actually have very much like our chytrids, have zoospores, but their zoospores, unlike the chytrid, are not flagellated. Now, common examples of blastocleidomycetes are water molds. They have that haplodiplonic life cycle, and their female gametes actually secrete a pheromone to attract male gametes, which I think is really kind of a fun thing. So we'll start off on 2N, as we always do. So we have this structure here, what we call the mesosporangium. It undergoes meiosis to produce haploid spores. In this case, they're just noted as X, which then develop into gametophytes, which then progress into young gametophytes, which then develop into a fully mature gametophyte, again, haploid. Now, you notice there's a female and a male gametophyte here, so they have a level of differentiation sexually. So instead of plus and minus, it's male and female. Now, this leads to the production of male and female gametes, which then fuse together, or I should say fertilize one another, or one fertilizes the other, I should say, leading to a sporophyte, which is 2N or 2X, which then leads to a young sporophyte, which leads to a mature sporophyte, which allows us to do one of two things: undergo asexual reproduction, producing more young sporophytes and more mature sporophytes, or producing those meiosporangium, which allows meiosis to occur, thus repeating the cycle over and over again. Now, again, the interesting thing about this is in their process of fertilization, a female secretes a pheromone to attract male gametes. So it's kind of like the way we think of like a mating call, right? They're secreting something in the environment, say, hey, I'm ready to be fertilized. Come over here. Let's get down to business, essentially. Now, our final organism is the I'm not going to try to say it. You can Google how to say it. I'm just not going to try. These are organisms that digest plant biomass in mammalian herbivore ruminants, so things like cows. They actually have a greatly reduced mitochondria that actually lack cristae. Now, you'll remember cristae are those basically folds that allow the electron transport chain to occur. They produce zoospores, which actually have multiple flagella. And the really interesting thing is they've actually have this really well-documented case that brought cellulase, which is a gene that breaks down cellulose from bacteria into this group of organisms. And the organism that was studied for this is actually Neocallimastix. So pretty cool. Now, the final group of fungi, which is sometimes associated with fungi, but not always associated with fungi, is the microsporidia. These are obligate intercellular animal parasites. Now, a long time ago, they were thought to be protists. Now they're more closely resembling fungi in terms of how scientists think about them, but we're still not quite sure. They do lack mitochondria, so they actually don't do aerobic respiration, but they are obligate parasites. They do rely heavily on their hosts for energy and things like that. Now, these organisms, including encephalozoan cuniculi, I believe that's how you say that. Again, you guys know at this point, I'm terrible at pronunciation, especially of hard to pronounce Latin words, but they commonly cause disease in those that are immunosuppressed. Now, interestingly enough, they infect their hosts with spores, which contain a polar tube. So you can see their spore here, this whole spore structure here, and they have these polar tubes. Now, these infect the intestinal as well as neuronal cells or brain cells, leading to diarrhea as well as neurodegenerative diseases. Their name tells you what they do, right? Encepha. What does encepha mean? Swelling of the brain. So basically, these polar tubes allow them to penetrate, infect the host, allowing them to proliferate as pathogens. Or technically, I should say parasites, but you could think of them interchangeably. So that's going to be it for our tour of the microscopic world of protists and fungi. I hope you did learn a lot about these groups. Very, very diverse group of organisms. They could be an entire course all on their own. But I tried to give you the broadest overview of what this group of organisms is capable of, some of their diversity, and all some of the really wild and wacky things that these organisms do. So as always, if you have any questions, please do let me know. But otherwise, I hope you guys are having a great day and take care. Goodbye.